So alternative mechanisms of carbon fixation have evolved in hot, arid, dry climate. Dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants close up stomata, which conserve the H2O but also limits photosynthesis. Why? The closing stomata reduces access to CO2, so CO2 cannot coming in through the stomata because it's closed up, and causes O2 to build up because of the light reaction. O2, they release out O2, 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 so O2 concentration increase up and cannot go out because of the stomata close up. So these conditions favor an apparently wasteful process called photorespiration. I want you to remember also what is photorespiration too. I want you to remember. In most plants, as I mentioned earlier, most of the plant, they fix carbon into 3 carbon molecules. So most of the plant, they are C3 plant, or they go through C3 photosynthesis because initial fixation of CO2 via Rubisco enzyme forms a 3 carbon compound, 3-PGA. In photorespiration, Rubisco adds O2 instead of CO2 in the Krebs cycle, producing a 2 carbon compound. Photorespiration consumes O2 and organic fuel and release CO2 without producing ATP or sugar. So by the photorespiration, Rubisco enzyme, not with CO2 but add up O2 and producing 2 carbon compound. Those 2 carbon compound gonna get broken down into CO2 and go away into the air. So organic compound, they go away. They missed out. That's the what? That's the photorespiration by Rubisco enzyme make binding with O2. That's the wasting process. So instead of gaining carbon, they losing carbon. That's the photorespiration. So it occurs in very hot, dry weather condition when stomata close up completely. In many plants, as I mentioned, most of the plants, they are C3 plants. Photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Krebs cycle. They wasting. But some plants, they reduce down photorespiration, like in C4 plant. C4 plant minimize the cost of photorespiration by incorporating CO2 into 4 carbon compound, not 3 carbon compound, but 4 carbon compound. That's why we call them as C4 plant initially. There are 2 distinct types of cell in the leaves of C4 plant. One, bundle sheath cells are arranged in tightly packed sheath cell around the vein of the leaf. Mesophyll cells are loosely packed between bundle sheaths and the leaf surface. This one typical anatomy for C4 plant. So it's showing a section of leaf of C4 plant. That's the mesophyll. Through the mesophyll tissue inside the leaf, we can see vein, vascular tissue to transport water and sugar. And surrounding the vein, we can see bundle sheath cell. So it protecting the vein tissue. And those bundle sheath cells, cell wall, they will build up cell wall. And with the cell wall, they producing a waxy material and they deposit inside the cell wall. So that's why it develop a thick cell wall. And CO2 and O2 cannot go through that cell wall of bundle sheath cell. That's the typical anatomy. And surrounding mesophyll cell inside the chloroplast, we cannot find the abundant enzyme Rubisco. Instead of Rubisco enzyme, we can find some other enzyme. That's the C4. So inside the mesophyll cell in chloroplast, for C4 plant, we can find enzyme PEP, carboxylase enzyme. So sugar production in C4 plant occurs in a three-step process. The production of the 4 carbon precursors is catalyzed by enzyme PEP carboxylase in the mesophyll cell. So, to the PEP carboxylase enzyme, C3 and CO2 bind into. Then that enzyme converts them into C4. That's why we call them as C4 plant. So PEP carboxylase has a higher affinity for CO2 than Rubisco enzyme does. It can fix CO2 even when CO2 concentrations are low. These 4 carbon compounds are exported to bundle sheath cell through the plasmodesmata. They're using plasmodesmata, one the cell to another, that C4, they are transported through the plasmodesmata, through the cell wall. And within the bundle sheath cell, those C4 compounds get broken into C3 back and CO2 released out. So they release CO2 that is then used in the Calvin cycle. So inside the bundle sheath cell, Calvin cycle is operating now. Now the CO2 with the Rubisco enzyme, they produce with C3 again, and regular Calvin cycle operating inside the bundle sheath cell to produce sugar, glucose, and other carbohydrate. So inside the C4 plant leaf, inside the mesophyll cell, with the PEP carboxylase enzyme, CO2 gonna get fixed into oxaloacetic acid, C4, and sometimes it can convert into malic acid, C4, and those C4 gonna get translocated into bundle sheath cell through the plasmodesmata, and inside the bundle sheath cell, the C4 gonna get broken back into C3 and CO2, and this CO2 with the Rubisco enzyme, and go through the Krebs cycle to produce sugar with regular, the Krebs cycle. That's the summary for the C4 pathway for photosynthesis. Therefore, the C4 plant, they can reduce down the photorespiration, especially in very hot, dry, and bright weather condition. C4 plant, they can grow up much faster than C3 in very hot, dry, and bright weather condition, like the corn plant, like the sugarcane plant, like sorghum. So some plants, they are classified as C4 plant. The other plant group, we call them as CAM plant. They borrow the name from plant family Crassulaceae. That's why CAM stands for crassulacean acid metabolism, CAM. So some plants, like the cacti, or pineapple, or jade plant, or some orchid plant, they belong into CAM plant. Those CAM plant, usually they live on very extreme environmental condition, very hot, dry desert they live on. That's why they cannot open up their stomata at daytime, because when they open up without sunlight, they're gonna dry out and get killed. That's why they wait until the nighttime to open stomata. Then CO2 gonna coming in. That CO2, using the PEP carboxylase enzyme, the CO2 gonna get fixed into C4 and store in the plant vacuole and wait until the daytime comes. So when daytime comes, they close up stomata, and that fixed C4 get broken down into C3 and CO2. That CO2 gonna get fixed with the Rubisco enzyme with the Krebs cycle to produce sugar in daytime with stomata closed up. That's why CAM plant open their stomata at night, incorporating CO2 into organic acid in C4. Stomata close during the day. Then those C4 gonna get broken and release out CO2. That CO2 gonna get fixed into C3 and other sugar by the Rubisco with Krebs cycle. That's for the CAM plant. That's why for the C4 plant and CAM plant, they show some similarity. C4 plant, the function for fixing into C4 and with the Krebs cycle, it is separated by the space of mesophyll tissue, mesophyll cell, and bundle sheath cell. So inside the bundle sheath cell, they produce sugar by the Krebs cycle. And for like the pineapple, for CAM plant, the fixation is operating in nighttime, at nighttime, and the fixed C4, they can wait until daytime. That C4 gonna get broken and release out CO2. That CO2 gonna get fixed into C3 and other sugar in daytime. And on dry, hot weather condition, C4 plant, the photosynthetic efficiency is very good, so they can grow very fast. But for the CAM plant, their major thing they're looking for, that's their survival. So maybe CAM, the photosynthesis, that's the strategy for their survival. So usually they are kind of slow grower. As I mentioned earlier, the photosynthesis is the greatest magic on the earth because it produces food for all the lives on the earth and the oxygen, O2, for respiration, for our survival. So the importance of photosynthesis is the energy entering chloroplast as sunlight gets stored as chemical energy in organic compounds. Sugar made in chloroplast supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. Plants store excessive sugar as starch in structures such as roots, tubers, seeds, and fruits. In addition to food production, photosynthesis produces the O2 oxygen in our atmosphere for our respiration. 네, 이번 챕터에서는 광합성, 지상 최대의 매직, 왜냐하면 우리가 손에 잡히지 않는, 우리가 눈으로만 볼 수 있는 태양 광선이 우리의 먹이뿐만 아니라 지구 거의 모든 생명체의 먹이를 제공해주는 광합성에 대해서 논의를 했습니다. 그래서 이 정도까지면 일반 생물 수준에 있어서는 충분하다라는 생각이 들어요. 일부 물론 교과서에 따라서 다르지만 일부 교과서에서는 광합성 바로 다음에 지구온난화 뭐 이렇게 논의를 하고 있는데, 지구온난화는 논의에서 좀 빼겠습니다. 논의하지 않겠어요. 그래서 왜냐하면 지구온난화는 물론 CO2 농도가 급격히 올라가면 기온이 올라가는 것은 맞아요. 하지만 지금의 대대적으로 정치적으로 떠드는 지구온난화는 사실은 지금 거대 기득권자들의 정치적인 논리에 따라서 굉장히 휘둘리고 있는 것이 사실이다. 그래서 논의에서 좀 빼겠습니다. 그래서 일부 정통 지구학자들의 견해로는 사실은 지금은 온난화 시기는 아니다. 차라리 빙하기의 초입에 접근하고 있다. 그렇게 주장하는 학자들도 꽤 많다. 그죠? 그래서 논의가 적절하지 않아서 이거는 지구온난화는 논외로 하고 지금 시기가 아직도 벌써 1년이 지나고 2년이 다 지나가는데도 아직 사회적으로 이 팬데믹이 안정화가 안 돼 있어서 어쨌든 그것도 놀랄거리가 많습니다만 어쨌든 따로 별도로 여러분들께 그 학기말고사에 대해서는 공지를 따로 드리도록 하겠습니다. 그래서 여러분들 시험 보기에 곤란하지 않도록 될 수 있으면 빠른 시기에 공지를 드리도록 하겠습니다. 그래서 비대면 강의로 저로서도 유감스럽고 미안하게 생각하고 여러분들도 비대면으로 강의를 듣게 돼서 힘드셨을 거로 생각합니다. 그래서 어쨌든 마지막 강의까지 여러분들이 들어주셔서 대단히 감사하고 학기말고사도 잘 보셔서 좋은 성적 받았으면 좋겠습니다. 그리고 여러분들의 모든 다른 학업도 좋은 결과가 나왔으면 좋겠고 늘 건강하시기 바랍니다. 이렇게 한 학기 동안 수고 많이 하셨습니다. 이만 하겠습니다. 고맙습니다.